chapter 31 in the part 1, we're going to talk about the innate host resistance. And sometimes this is also called as the innate host immunity or innate host defense system. There are several ways that we actually talk about that. So this video of the chapter 31 will be divided in two parts. In a part 1, we are going to discuss the section 31.1 through 4 and 31.5 through 7 we are going to discuss in part 2, as it is a large chapter. So in the first, we are going to define some of the terms and differentiate between the innate and the adaptive immune system. So first, let's take a look at some of the basic definitions that we will encounter in this chapter, as well as the next chapter, which is the adaptive host defense mechanism in chapter 32. So the first, the pathogen. What are pathogens? And we are talking about microbiology, and we are talking about all these pathogens all the time. These are disease-causing microbes. And interestingly, if we are talking about all the different types of microorganisms, actually the human pathogens or the pathogen of animals, plants are actually in a number less than the harmless microorganisms. So just small number of microorganisms that exist in nature are actually pathogen. And all of these must overcome the surface barriers and the antimicrobial activity. And here we are talking about mainly about the primates, human, higher vertebrates immune system, particularly in the human beings, where we see the most developed immune system. And what is an immune system? It is composed of a widely distributed proteins, cells, tissues, and organs, and their primary function is to neutralize or destroy the foreign substances, as well as essentially the pathogens. And the word immunity basically describes the ability of the host, which is the animal or the organism that will host or may host a disease-causing pathogen, is referred to as a host, where the disease-causing pathogen can replicate and use it as a host. And it is the ability of the host to resist an infection or disease. The reason for this, as we all know, that we exist in nature in equilibrium with all these pathogens, and we are exposed to all these pathogens in our everyday life constantly. So our immune system is the one which actually protects us against all those different types of pathogens, as well as foreign substances, which are inanimate object. And particularly the allergic reactions are the part of the immune system as well, which could be the foreign substances when alert our immune system or activate our immune system. And immunology is a study of immune responses, that how they protect basically the host. So here, an overview of the immune system, that you can see that this actually is kind of like the barriers. Like you can just think about that. These are some of the pillars or brick walls of the barriers, and this is a pathogen. Could be a virus, could be a fungus, could be any microorganisms, any pathogen. And the first thing that they have to breach is an anatomical and chemical barriers. And they are the part of our immune system all the time. And we're going to talk about these mucus, saliva, stomach, acid, and all those things we are going to talk about. Once this anatomical and chemical barrier is actually reached, there is a tier which we call as an intrinsic barrier. And these actually are the part, particularly for the viruses, it's the interferons, then autophagy, apoptosis, which is killing of the infected cells. And there are some of the other ones, which is the microRNAs are present. CRISPR is not in the human beings, but this is usually the defense in the bacteria. But these are some of the intrinsic controls. After that barrier is reached, a few of the pathogens will be able to do that. We do have innate immunity. Now, we are talking about the innate immunity, and just remember that all of them right here is basically, we're going to talk from anatomical to intrinsic to innate. We are all going to talk about these all three pillars within the innate host resistance. And the natural killer cells, the complement system, antigen presenting cells, neutrophils, these are just a few. But all of the other cells, which are granulocytes, like the dendritic cells, then you have the basophils, eosinophils. We are going to see that. Then the lymphoid progenitor cells. There are many different type of cells also associated with that, which are not mentioned in here. And these are all the part of innate immunity or defense. And we're going to talk about that entire part in this chapter. And then the next part we call is an acquired immunity. Why we call this acquired immunity, which is also known as the adaptive immunity, because this is very specific. Up to the innate immunity, this is a non-specific response against a pathogen, which could be many different types. Once it takes usually days, 3 to 7 days, it takes sometimes, a specific adaptive immunity develops, which specifically is designed to that particular pathogen. That means that it can actually act on a specific strain of a bacteria within a genus species, which does have a very specific character. It could be very specific to a specific type of virus, so it becomes acquired. That means that once the pathogen is observed by the immune system, it took a little time to develop that, or it adapted to that specific pathogen, and then it delivers its outcome, which we are going to see is a cell-mediated killing and antibody response. And interestingly, what happens is adaptive or acquired immunity does have the memory, which the innate immunity or the non-specific immunity doesn't have that. So this is the overall differences in between that. So again, innate immunity is a first line of defense, a first resistant to any microbe or foreign material. General mechanisms, we already talked about these: skin, mucus, antimicrobial chemicals. And the adaptive memory is tailored to a particular foreign agent. That is the important thing, activated by the cells and chemicals of the innate immunity. So essentially, the innate immunity trigger first, and the innate immunity, if it cannot contain the disease or the pathogen, it will constantly basically triggering or constantly informing the adaptive or acquired immune response. And it will activate some of the cells and the chemicals of the innate immunity, and it will be tailored to a particular foreign agent or a pathogen, and does have that memory. So now let's define a term antigen. So an antigen in any substance recognized by the immune system. And these are recognized by specific receptors that are found on specific cells. And the immune system can differentiate between a self-antigen and a non-self, which is a foreign antigen. So essentially, what is a self-antigen? So any molecule throughout our body, these are hundreds and thousands of different molecules, which could be a part of our cell, part of our body, which are could be peptide, proteins, carbohydrates, lipid, and glycosylated moieties. All these different chemicals and entities which are found in our body are called as a self-antigen. And our immune system is actually trained not to attack those or understand that these are the part of our self. On the other hand, there are many which are the characteristic of some of the antigens, and these antigens or entities are only found on specific pathogens which are foreign to us. Either the pathogen or could be a foreign material that could be, say, for example, allergy-inducing pollen may have a protein structure that is not present in our body, not self. That would be a foreign antigen. So this enables the immune system to rid the host of foreign invaders, as well as eliminate the cancer cells, because some of the cancer cells actually develop some of these markers which make them non-self. And this is the immune system can do it. Of course, there is a failure, and that's the reason cancer actually happens. But the immune system does have ability to reduce the number of cancerous cells which can show abnormal proteins on their surfaces. And when the discrimination goes away, it is a basis of allergic response and autoimmune disease as well. So we're going to talk about that as well. So let's take a look at the innate resistance, and it starts with the barriers. We are going to talk about this, the physical, mechanical, chemical barriers, and anatomy secretion. We are going to first talk about that. So essentially, this will be this first pillar. So in this first pillar, the barriers of the innate resistance is the host flushing mechanism, and these are the first line of defense. So constantly the sweat, urine, these do have the flushing effect, and we can just take a look at the different types of like you have the sebaceous glands, which contain these fatty acids, and that actually inhibits bacterial growth. Tears have the lysozyme. The mucus, saliva does have the lysozyme, lactoferrin and peroxidase. So the lysozymes will destroy bacteria. Lactoferrin actually take up iron, and they will not be able to deliver the iron, and all these different ones. And we are going to talk about each one of them at a time. So let's take a look at that. We first start with the skin. So the skin basically made up of stratified or cornified, converted to keratin epithelial cells. So the top layer of the skin is actually dead. So if you have a thin skin, you have 30 to 50 layers of the dead cells, and these cells do not lose their integrity. They maintain the shape, but they are essentially dead, and they are filled up with these abrasion resistant protein, which is called the keratin. And as we go deeper, we have the other layers, stratum granulosum, spinosum, and basale. So this stratum basale is the deepermost layer of the epidermis, and this is the only dividing part of the epidermis. So this is the cell that divides, and underneath that you have the dermis, which does have the blood vessels. And the nutrients diffuse from the dermis to this epidermis, and the waste product are diffused out. So as the cells of the stratum basale actually divide, the older cells are pushed upward, and they gradually, you can see from this cuboidal shape, they become kind of stratified and squamous. So they become like a fish scale like cells. They flatten out as they move upward. And then there is a critical area when they reached beyond the layer of the stratum granulosum. They are too distant to receive nutrients and eliminate waste products from the underlying dermis, and they gradually die. But before that, there is a huge change that happens in these cells, that they actually accumulate this keratin in fibrous fibrous protein, and they accumulate in the cell, and that becomes the abrasion resistant layer. And you can see that actually gives the skin its barrier, and the microbes cannot easily invade that. So on the other hand, just think about that when we do have a cut in the skin, or we have braided the skin, so this part of the skin is gone, or in a burn patient, this part of the skin is gone. We are exposed. We expose the deeper layer of our skin, and we are more prone to infection. So these cells are called keratinocytes, which essentially are rich in keratin, and essentially this prevents the microbiota to prevent colonization of the pathogens. So we are also going to see that some of the cells, which are the cells called Langerhans cells, these are some of the cells which essentially roam around and move around in the tissue, particularly in the epidermis, and they are responsible for catching any pathogen and deliver it to the immune system. It's required. We're going to talk about that as well. So on top of the surface of that, the different fatty acids that are also removed make the survival of the microbes a little difficult on the skin surface. So after that, we are talking about the mucous membrane, and all our bodily openings are protected by the mucous membrane, as well as our internal lining of our gastrointestinal tract and many other areas. They do have the mucous membrane, and they form a protective covering that resists the penetration and traps the microbial invaders. And what are the different types? That these bodily openings do have these epithelial cells, and in between these epithelial cells, you have a certain type of cells which are called the goblet cells, and they produce a polysaccharide which is known as a mucin, MUCIN, and it mixes with the water and produce the mucus. So mucin plus water becomes the mucus. And also, there are some specific cells. They are found in the deep crypts of the intestine, which are called the Paneth cells. And they also secrete the antimicrobial peptide, and they keep our gastrointestinal tract infection free. And also, there are mucosal secretion that is in tears, and these are also toxic to the microorganism. Say, for an example, that the tears do contain these the lysozyme, and the lysozymes basically destroy the peptidoglycan layers and lyse the bacterial cells. Now let's take a look at the respiratory system and how it is protected. So the turbulent airflow essentially can deposit the microbes on the mucosal surface, and the microbes greater than 10 micron usually are trapped by the hairs and cilia lining of the nasal cavity. But if they are lesser than that, and most of the bacteria are, pass through the nasal cavity, and they are trapped in this area, and we call this as a mucociliary escalator, where you are seeing a pseudo-colored electron micrograph of this. So these are on the right-hand side are the columnar cells that you were saying that this is the epithelial cells. The goblet cells are also within those. And what happened that these epithelial cells, these are particularly pseudostratified. We call this a pseudostratified epithelium, particularly in the respiratory tract, and they contain all these cilia. And along with that, there are interspersed goblet cells that secrete the mucus, and this entire surface area of the mucus of the cilia, they are covered in the mucus. And what happens that these cilia actually move in a particular way from downward to upward motion, and that moves the particles away from the lungs towards the mouth. And then coughing and sneezing also clear the system. So why we call it a mucociliary escalator? Because they are constantly, these cilia beat upwards, and they have a constant upward movement of the trap microorganisms or foreign substances which move towards the mouth. So sometimes what happens, then this is something related to that. Sometimes when we drink something cold, it soothes our mouth as well as throat. But sometimes that during an infection or something when you are coming with, we are told to gargle with hot water, salt, or those kind of things, and avoid cold water or the cold substances as well. And there's a reason for that. So basically what happens that when we usually take cold water or ice, the movement of this mucociliary escalator actually slows down, because any enzymatic reactions or cellular response will slow down in a lower temperature. So when we usually drink hot soup or tea or take a steam or gargle with warm water, this actually makes these mucociliary escalator more active to push the pathogens upward towards the mouth so that they can be destroyed. So this is the story behind these warm food gargling, those kind of things, that why it actually helps. Also, along with that there are phagocytic cells in the lung, and we call these as alveolar macrophages. We are going to talk about these. These macrophages are produced as monocytes in the blood. They leave the blood vessels and then wander around in the tissue, and they mature into macrophages. And they can phagocytose directly the different bacteria, which cannot be cleared by the cilia or the mucociliary escalator. So after that, we are talking about the gastrointestinal tract defense, and the stomach encounters many microorganisms, and the defense system in the stomach is extremely strong because it contains the hydrochloric acid pH almost at the range of pH 1 in the stomach. It contains proteolytic enzymes as well as the mucus, and all of them actually protect us. But there are some of the microorganisms that can actually withstand those. So there are some viruses which can withstand, say, for some of the stomach viruses like rotaviruses, noroviruses, they can withstand that stomach, so the extreme stomach condition. On the other hand, there is one bacteria which is Helicobacter pylori, which is one of the main causal organism of the stomach ulcers, basically, that actually can withstand this acidic secretion. And it's a very interesting way that how Helicobacter pylori actually basically manage this situation in this. The Helicobacter pylori actually embed itself in the mucus layer of the stomach, and they actually secrete a neutralizing alkaline solution around them, and around them they make a microenvironment which is not acidic. So they make a neutral microenvironment around each the cells being embedded in the mucus, and that's why they can fight those. But most of the microorganisms will be destroyed by these extreme conditions. But still, some of the microbes can get passed into the small intestine, not destroyed by those. And for those, there are pancreatic enzymes. Then the bile. So the bile is basically the saponification or emulsification agent. So think about that. The bile salts are just like detergents or soaps. And those are these molecules, like, say, for example, deoxycholate is like a sodium deoxycholate. These are some of the bile salts. They are amphipathic. That means that they have a part which is hydrophobic, one part that are hydrophilic. And these essentially can embed into the membrane of the envelope viruses, as well as bacterial membrane or the organisms, and then basically lyse them. So they can basically are the essential role of the bile is essentially for the fat digestion. So it emulsifies or saponifies the lipid in the food, which is it's basically like breaks up chunks of lipids into like a water miscible kind of emulsion, essentially, so that the lipases or the enzymes can actually access the lipid for digestion much more easily. But they also are can help in destroying the lipid phospholipid bilayer of some of these microorganisms which can pass into them. So intestinal enzymes, of course, in there. And then there is a particular area we called as a GALT, or gut-associated lymphoid tissue. And there are certain areas or patches around that. We are going to talk about that, like Pear's patches. And these are particular areas which are rich in different types of cells. And there are some certain kind of cells which actually constantly, and one of them is called the M cells. And these M cells, and again, but not detailed, not necessary here, but these M cells actually send their long arm, which are like tentacles, inside the small intestine and actually check for microorganisms. And if they actually, the M cells in the GALT area, which are in the GALT, actually catch the microorganisms, they will inform the cells underneath them, which are the B cells, T cells, which are the part of the adaptive or the acquired immune response. They will inform them and they will come into the action as well. So there is constant monitoring happens in the GALT, which is the gut-associated lymphoid tissue. And along with that, the peristalsis and basically moves some of these cells which are lining the intestine. So the intestine is essentially lined by these epithelial cells, which are responsible for the movement of the food. And of course, in the long part of the intestine, they are responsible for absorptive functions. They absorb, but they are constantly replaced. So they are constantly replaced. So if some of the cells are infected, by in the process of purging them out, the microorganisms and the infected cells are purged out as well. But the gut actually contains a very healthy microbiome, and this microbiome is essentially our own flora, or which is a part of the bacteria, fungus, and now we just probably trying to think that maybe some of the viruses are also present in there. And all this microbiome plays an important role in the immune system. And many of these individual agents, microbiota, actually prevent the growth of potential pathogens. And we are going to take a look at those in the later part. The next one is the genitourinary tract, and this is unfavorable environment for foreign microbes. So the flushing action of urine actually flush the microbes away. Low pH of the urine due to the uric acid, that is also helpful. And the urea, other metabolic products, which are also detrimental for the growth of microorganisms. So now what happens that the male urethra is about 20 centimeter long compared to about say 5 centimeter for the female, and that's one of the region that due to the anatomical differences in between the male female urethra, what happens that the females are more prone to urinary tract infection, about 4 times more prone to urinary tract infection. But on the other hand, what happens that there is an innate defense for the vagina because it is opened outside, and the normal microbiota basically, which is dominated by lactobacillus, particularly generate an acidic environment in non-pregnant women as well. And then what happens that why we actually, as part of the textbook, we say that non-pregnant women, because what happens that after pregnancy, the lactobacillus species diversify and there are many others. So essentially there is a shift in the microbiota or the vaginal microbiota in pregnant and non-pregnant women. But these bacteria actually provide an acidic environment and a protective environment as well. And of course, there is the mucous membrane. So when there is a mucous membrane, there is always several agents in the mucus which are constantly making this. See that right here, if we're just looking at that, the mucus also contains the lysozyme. It also contains these lactoferrin, the peroxidase, as well as transferrin. These are some of the things that are present in the mucus, and those actually help the growth of the microorganism as well. So after that, we are looking at the innate resistant, and we are talking about the chemical mediators. And we are going to talk about their microbial action, general terms of activation, and we are going to talk about some of the cytokines and their major functions. So the first thing that what we are looking at is a chemical mediator of innate resistance. It's essentially it's host a chemical arsenal with which we can combat onslaught this microorganism. The first one is the hydrolysis, the bacterial cell wall is the lysozyme. So you can see that this is basically your peptidoglycan. You have an acetylglucosamine and acetylmuramic acid, and this is essentially broken by the lysozyme. And there are two molecules, which is a lactoferrin and the transferrin in the blood. And these lactoferrin and transferrin basically sequester or capture iron, and that is not available to the microbes and limiting their ability to multiply, because the iron is absolutely essential for the electron transport chain, just like us, even for the microbes which are doing the different type of metabolic activity. So many different pathways require iron, and sequestering the iron actually limit their growth. So after that, we are talking about a group of cationic peptides, and there are 3 classes of them. So these are all biologically active cationic peptides, and they can damage the bacterial plasma membrane, mainly the cathepsyllin and the defensin. And the histatin is a little bit different. So these cathepsylidins, these are alpha helical peptides, and usually these are produced as a large protein and then proteolytically processed as cathepsylidins. And what they do that they can embed themselves into the membranes, which include the bacterial plasma membrane or the lipopolysaccharide layer or the envelopes of the enveloped viruses, and then they can disrupt those membranes. So that's the function of the cathepsylidins. On the other hand, the defensins, which is the alpha and the beta defensins, and the alpha defensins are smaller, and these are also processed. Both the defensive are also produced as a larger protein product and processed. And these are smaller, found in granules of neutrophils, intestinal Paneth cells, and intestinal and respiratory epithelial cells. And the beta defensives are found in the epithelial cells and stimulate the release of the chemical mediators as well. So two different types of a function. The main difference between the cathecellidins and the defensives are the cathecellidins are found in the vertebrates only. On the other hand, the defensins are found in the vertebrates as well as invertebrates. Histatins, on the other hand, are larger peptides found in the saliva, and they compromise the mitochondrial function, and they generate oxidative and osmotic stress, resulting in the fungal death. So this is, in terms of that, is a little bit different from the cathecellidins and the defensins, which essentially attack the membranes. The next are the bacteriocins, and these are also peptides, protein product, and they are produced by the normal microbiota, and these are developed due to the competition and kill closely related species. And the colicins are basically produced by the E. coli, and they bind a receptor on the cell envelope of sensitive target bacteria and cause the cell lysis. And one group of these bacteriocins are called the lentibiotics. These are produced by the Streptococcus, Bacillus, Lactococcus, Staphylococcus, and many of them are the natural microbiota of our own. The next one is the complement system, and this complement system is pretty complex, and it is composed of about more than 30 heat labile proteins. And this essentially complements the antimicrobial activity of the immune system. And they are present in the blood serum. So if we just basically draw blood in a good amount, and then if we just let the blood cells coagulate and precipitate, and if we just get that yellowish straw-colored part of the blood, which we call as a serum, we will find these complement proteins along with those as well from those serum. So essentially, they stimulate an inflammatory response by helping to recruit the white blood cells, leukocyte. They lyse microbial cell membrane, and they also enhance the process of phagocytosis of microbial invaders. And a particular word is used for that, which is known as opsonization, which is promotion of phagocytosis or enhancement of phagocytosis. So three major things. So these complement proteins actually circulate in the blood in an inactive form. And if there is a microbial invasion, there is an activation of the complement system, and the activation can happen by three different mechanisms, and we are going to talk about each one of them. So first, let's take a look at that what is opsonization, or the promotion of phagocytosis. So the microbes can be coated with the complement proteins, antibodies, or both. And the opsonin are basically the proteins that coat these microorganisms. And this promotes the recognition by phagocytic cells. So let's take a look at that, how these opsonization works. So here we are looking at a phagocytic cells, which could be a neutrophil or our macrophage. Here is a microbe. And this microbes does have a specific protein or signature on the surface, which can be recognized by an antibody. And we did not talk about antibody still yet. That's a part of our adaptive immune response in the next part. But just a basic idea that these are secreted by our immune system cell, particularly adaptive immunity. And if they coat the bacteria with this antibody, this Fc receptor, which is the stalk portion of the antibody, does have a specific receptor present on the phagocytic cell, and this can enhance the degree of binding and can enhance. So this is the antibody-mediated phagocytosis. Now, there's another way that there is a particular protein, which is the C3b. C3b is a complement protein. And these can bind to the microbes, and this can be recognized by a C3b receptor present on the phagocytic cell. And if these two interaction happens, the degree of binding or opsonization increases. On the other hand, if a microbe is coated by an antibody and coated by the C3b, we get the highest amount of opsonization, or this is the most effective triggering of the of the phagocytic process. So the degree of, depending upon that what is present on the microbe, the degree of phagocytosis can be greatly enhanced. Antibody and complement C3b, we see the highest amount of opsonization or degree of phagocytosis. So here we are looking at the three different parts of the complement, and there are three main parts of these complement system activation. One is a classical pathway, and when antigen antibody complex is formed, this classical pathway is triggered. So here we are looking at a bacteria, and this Y-shaped molecule is an antibody that we saw in the first place right here. If this is actually happens, and if the complement proteins actually recognize that, this will trigger the classical pathway, which will be a cascade of reaction, which essentially will trigger one protein, the next, then next. And so this activation of this cascade will actually happen. So this is your classical pathway. And then there is the next one. What happens is that this is your mannose. Mannose, these are sugar, which are mannose. And the lectins are essentially proteins which bind to carbohydrates or sugar. So lectin is a general name of all the carbohydrate binding proteins. So these are mannose binding proteins, can actually essentially recognize those. And when the mannose binding proteins actually recognize these mannose sugars present particularly on these microbes, they essentially activate the lectin pathway. And then there is an alternative pathway, and this is activated by the lipopolysaccharides and different types of surface antigens. And all of these essentially, all the pathways there, it is much more complex, but just an overview is good enough, that all of them actually come to a common thing, which we call as a C3 convertase part. So C3 is a complement protein 3, and this is a C3 convertase. Now this C3 is divided into two parts, or cleaved into two parts. One is a C3A, and the C3A is a potent trigger for inflammation. On the other hand, this C3B, we already saw this, is a potent opsonization agent which promotes phagocytosis. Now C3B also make the C5 cleave into two parts, C5A. C5A again is a promoter of inflammation, and the C5B essentially after that will recruit the complement protein C6 through 9. So C5B, then C6 through 9, and all these protein complexes actually make a pore, which we call as a MSC, or membrane attack complex, and these are inserted into the bacterial membrane and lyse the membrane. So these are the three outcomes of those: stimulate inflammatory response by helping to recruit white blood cells, leukocyte. This is done by the C3A and C5A. Lysing microbial cell membrane, this is by the membrane attack complex. And the C3B promote the phagocytosis by opsonization. These are the three major activities that is shown: phagocytosis, membrane attack complex, inflammation, right here. So this is the alternative pathway. When the pathogen has an LPS surface antigen, this pathway is initiated. This begins with the activation of C3 to cleave itself into this C3B as an opsonin, and acts as a protease in a complex of other proteins that we just talked about. And that essentially recruits the C5B and then C6, 7, 7, 8, 9. And all these proteins, one after another, one is C5A, then you have a C6, you have a C7, then many molecules of these C9, they are the similar. They all recruit together to form this round complex. And here you can see in an electron micrograph, these are essentially the pores that you can see, and see the material from inside the bacteria is essentially being extruded out. So they can basically lyse the gram-negative bacteria's lipopolysaccharide layer by making this membrane attack complex. They can also attack the enveloped viral membrane as well. So lectin pathway, again, we already talked about this activation of the C3 lectin binding. This depends upon the interaction of the mannose binding lectin proteins. And the complement pathway is basically for the antibody specific to the pathogen binding. And this antibody was produced by the activated B cells as a part of an adaptive immune response. So this part will make complete sense when we talk about this in the next chapter. So here is the overview of a little bit more detail, the activation of the C3 convertase, which is the heart of the complement system. So here you can see in the classical pathway, and detail of these individual factors or names are not needed, but just showing that how the cascade reaction happens. So here, a protein called C1q binds to these antibodies, which is cleaved into C1r and S. That activates the C4, C2, that will be converted into C4b and C2a, and this is your C3 convertase. And this can be produced by this activation of mannose binding lectin and MASP as well. So the MASP is essentially the mannose binding protein serine protease. So this is a protease which will essentially make these act on these proteins and make these C3 convertase. On the other hand, see that right here, the alternative pathway when LPS or surface antigen activate this C3b, there are factor B and factor D. Those basically will associate with that and also make a C3b on BB C3 convertase. And once these C3 convertase, which are essentially proteases, act on the C3 complement protein 3, it cleaves it into two parts, which is C3a and C3b. And see, the C3b essentially activates this pathway. So it feedbacks into the loop because it's necessary with that. So C3 is essentially the heart of the complement system. No matter which pathway it is, if the C3 is essentially activated, that will essentially do everything else. It can do the inflammation, it can opsonize, and it can help in the membrane attack complex formation. So again, the textbook actually is very detailed. You have all the names of all these factors and their individual functions, but that details in between the lines is beyond that detail is not necessary. As long as you have the overview of the pathways, that is good enough for the scope of this course. So after that, we are looking at the cytokine activity. So the soluble proteins that actually initiate the signal transduction pathway that regulates transcription of immune response gene. And these are the different categories: regulators of innate immunity, adaptive immunity, and also stimulators of hematopoiesis. That means that production of the blood cells. The reason for that, of course, the hematopoiesis also produce red blood cells, but mainly we are talking about the cells which are the white blood cells, and these white blood cells actually produce a number of different types of cells of the immune systems. So let's take a look at the different cytokine functional groups. So the first one, these are called the chemokines. These actually stimulate the cell migration. Kine means movement, and this stimulate the cell migration. So if there is a tissue which is invaded by a pathogen or it is injured, the local cells actually release some of these chemokines, and these chemokines actually attract the immune cells to that site. And we are going to talk about that in the much later part in this chapter as well. Then the interleukins are produced by one leukocyte and act on another. So what will happen, the chemokines will stimulate the cell migration, and many of these different leukocytes of the white blood cells will reach the site of infection or injury. And at that time, those leukocytes will understand what's going on, and they will release these interleukins, which will activate others. On the other hand, the interferons are regulatory cytokines produced in response to a viral infection, and this is very specific. Interferons, there are several different classes, and they are released upon viral infection only. And very interestingly, what can happen, the interferons are basically processed through the different pathways, and these are transcription factors, and this can stimulate what we call as an interferon stimulated genes, or ISGs. And it is kind of mind-boggling to understand that so far, more than 1,100 genes, which are the protein products, have been identified which can be turned on into the interferons. So when a particular cell is infected by the interferon, when a particular cell is infected by a virus, it releases or produces interferons. These are secreted outside the cell, and these interferons essentially is bound to the receptors on the neighboring cells. And all the neighboring cells which are close to that basically turn on the ISGs or the interferon stimulated genes and produce these antiviral proteins and make those cells into, or they try to make those cells immune to the viral attack, or we call this make those cells in an antiviral state. And of course, viruses can actually thwart that response, of course. But that is the function of the interferon, to make these wide range of antiviral proteins, which can basically stop the viral infection into the neighboring tissues. The next group of cytokine is a colony stimulating factor, or CSF, that stimulate the growth and differentiation of immature leukocytes in the bone marrow. And these are essentially the white blood cells, the leukocytes. And then the leukocytes, essentially, we're going to see, are mainly going to develop into what we call as a granulocytes and agranulocytes. And then there are another group of white blood cells, which we call as a lymphocytes. The lymphocytes give rise to the T cells, B cells, and T cells and the B cells are the part of the acquired immune response. But there is also another type, which is called the natural killer cells, is a part of our innate immune system. And there are some of the cells of the same lineage, which are not leukocytes, they are lymphocyte lineage. They are also responsible for carrying out different functions in the innate immune system. And we are going to talk about that in a little bit later part. And also another part is a tumor necrosis factor, or TNF. And the TNF basically stimulate an inflammatory response. So essentially what happens is that sometimes this immunity, we call this, is like too much of a good thing. So when your tumor necrosis factor happens, the inflammatory response is essentially what happens, that there is a vasodilation. So that area actually become inflamed, and the blood capillaries actually they become enlarged. So more blood cells can rush to the site, and the cell migration can actually happen. But due to the larger volume of blood coming to that tissue, the tissue actually retains water and actually swell. And that actually causes the reddening of the site due to the increased amount of blood flow. But the swelling actually happen due to the fluid retention in that site. So these are the classic inflammatory response. But sometimes those are uncomfortable and detrimental to the body as well. So these are some of the parts of the part of the cytokine groups and their function. So after that, there are some acute phase proteins, and these acute phase proteins are essentially the macrophage activation actually leads to that. So macrophage activation will cause this cytokine release, and this will stimulate the hepatocyte to produce these acute phase protein production. And this assists in the prevention of blood loss and ready the host for microbial invasion. So this actually contains the C-reactive protein, we call the CRP, then MBL, which is a mannose binding lectins, surfactant proteins like SPA and D, which bind the bacterial surfaces and act as an opsonine. And also, along with that, there are other proteins like colectins, which are the collagen-containing C-type lectins. And this family of collagenous calcium-dependent defense lectins, they can bind to the surface of the microorganism between the carbohydrate, triggering aggregation and opsonization. And this is essentially the response to the sites of the injury. All right, so after that, we are looking at some of the immune cells, and each one of them have a specific function. So let's take a look at those. And the cells of the immune system are leukocytes, white blood cells of the immune system. and all originate from the hematopoietic precursor stem cells. And mast cells, granulocyte, monocyte, macrophages, dendritic cells, and then innate lymphoid cells that we just talked about. So let's take a look at this entire part. So here you can easily see that this is the original hematopoietic stem cells in the bone marrow. And these cells, these are the original stem cells, uh, uh, differentiate into two types. One we call as a common myeloid progenitor, and another becomes uncommon lymphoid progenitor. So we will go to mainly the common myeloid progenitor because these cells are responsible for the innate immune system related cells. But just quickly, that we will talk about these in the adaptive immune response, except a few. Let's quickly talk about this the lymphoid progenitor. So the lymphoid progenitor can produce these lymphoblasts, which produce the T cells and the B cells. These T cells and the B cells, these are the part of the adaptive or the acquired immune response that we are going to talk about in chapter 32. But other than that, there are few cells like these cells. These are the part of the innate immune response, and these are being, so we're going to talk about these individually later. So one of them is a lymphoid tissue inducer cells. Another is innate lymphoid cells, type 1, 2, 3. We are going to talk about their individual functions, what they do, and the natural killer cells, NK cells. These are the integral part of the immune system. So these three are the part of the innate immunity, and we're going to talk about it. Other than that, the common myeloid progenitor, they differentiate into several different things, and two of them are not the part of the immune system. One of them is erythroblast. From there, it produces the red blood cells, RBC. On the other hand, the megakaryoblast becomes megakaryocytes, and they are fragmented to form the thrombocytes or the platelet involved in the blood clotting and inflammation. So these are essentially have other function. Now these are the one which are mainly responsible for the most of the cells. So one of them is what we call as a putative mast cell precursor and produce a mast cell. These are the specialized cells that we are going to talk about, responsible for the allergy. Then also it produces what we call as a dendritic cells. These are cells like star-shaped with long arms, and they actually move in different tissues to capture microorganisms. And one of the examples that we talked about that is your Langerhans cells in the skin. Those are basically dendritic cells. They capture the pathogen and bring them to the lymphoid tissues to present them. We're going to talk about that. And then this myeloblast, these cells essentially produce three granulocytes. These granulocytes are eosinophil, basophil, and a neutrophil. And the way they are basically essentially labeled is the way that their granules, they have these all different enzymatic granules in their cytoplasm, and how they are stained based on those. The neutrophils have a kind of a neutral granules, basophil basic granules, eosinophil has an acidic granules, and that's how the hematoxylin eosin stain them, and that's why they are named like this. And we're going to see that the function of eosinophil, particularly in fungal infection, allergy, inflammatory reaction, and parasitic infection. Basophil in allergy, inflammatory reactions. Neutrophil is a very, very important phagocyte, and we're going to talk about that, and important for bacterial infection. On the other hand, the common myeloid progenitor also differentiates into monoblast, and that gives a monocyte. This is an agranulocyte. This monocyte, just like the lymphocyte, doesn't have a granular cytoplasm like the eosinophil, basophil, or neutrophil. Now, this monocyte is a blood phagocyte that essentially circulate into the blood just for about 8 to 10 hours. And they leave the blood vessels into the tissue space, and they basically mature into the macrophages. These are the largest phagocytes that ingest, kill foreign cells, strategic participant in certain specific immune reactions. So these are the main players of the immune system. So again, repeating that, what are the main players of the immune system? You have the eosinophil, basophil, neutrophil, monocyte, macrophages, dendritic cell, mast cell, natural killer cell, and also innate lymphoid cells, different types, and the lymphoid tissue progenitor cells. These are the part of the innate system. So let's take a look at one thing at a time. So the mast cells are, we already talked about that, bone marrow derived, differentiate into the connective tissue. These are non-phagocytic. And there is a process called degranulation, is the process of release of the granules in the extracellular environment. So the mast cell contain many of these vasoactive mediators in their granules. And what are these? These are histamine, prostaglandins, serotonin, heparin, dopamine, platelet activating factor, leukotrienes. So what we are essentially seeing, this is an electron micrograph of a rat mast cell. And these are all the granules that we talked about. And these are the vasoactive mediators. On the surface, the mast cells do have a receptor, which is an immunoglobulin E type. We call this an IgE, which is not required. We will learn that in the next part. So when a particular cognate molecule binds to the immunoglobulin E, IgE, it can stimulate the mast cells. And this is the process of the degranulation, which is the release of the granule in the extracellular environment. When the degranulation happens upon activation of the mast cell, this will play the most important role of the allergy. So now we know that this is responsible for this. And very interestingly, what happens that when we take the anti-allergy medicine, which is a Zyrtec, Claritin, Benadryl, that essentially doesn't allow the degranulation process of the mast cell. It keeps the granules inside. So even when the activators bind to the IgE molecules, the process of degranulation is prevented by the allergy medication. So we do not get those allergy responses and inflammatory responses. The next ones are the granulocytes. We already talked about each one of them. So let's talk about that individual one of them. So basophil, irregularly shaped nuclei with two lobes, contain the histamine and other vasoactive mediators. So this is also for the histamine is, of course, allergy vasoactive mediator that will cause the inflammation, which is rending of the eyes, sneezing, coughing, and itchiness. All these are due to the release of the histamine. So the allergic responses also will basically contain the basophil as well to some extent, but they mainly play the role on the mast cells and play important role in the development of allergy and also the hypersensitivity. So this is your basophil, one of the type of the granulocyte. The next one is the eosinophil, and multi-lobe nucleus, two-lobe nucleus collected by a slender thread of chromatin and contain hydrolytic enzyme in addition to peroxidases and major basic proteins. So essentially what happens that they circulate in low numbers, migrate to the bloodstream and the tissues, and migrate to mucous membrane when recruited by the chemokines. And they are important for the protozoan and the helminth parasite. And they release these hydrolytic enzyme, cationic peptides, reactive oxygen species, because they have these peroxidases, and the damage the parasite's plasma membrane also produce this reactive oxygen species. So basically that will be damaging to the membrane proteins as well, and also play a role in the allergic reaction. So this is the other part of your innate immune response, other part of your innate immune response, which are eosinophil. The neutrophil, usually 3 to 5 lobes connected by chromatin, highly phagocytic, and this circulate in the blood and then migrate to the sites of the tissue damage, usually just like the macrophage. And these are the most important one in bacterial infection, and they kill the phagocytose microbes with and also hydrolytic enzymes and reactive oxygen metabolite contained in the primary secondary granules, which essentially kill the microbes after phagocytosis. So very, very essential in bacterial infection. So many times that we talk about some of these that the patients, particularly who are getting radiation therapies and also on immunosuppressive drug, they actually suffer from a condition which is called the neutropenia. Now, the thing is that what happens that in the blood, the number of basophils or the eosinophils is very, very low, by far very low. On the other hand, the most common leukocytes or the white blood cells that you have in the blood is the neutrophil. So what happens that particularly for the patients who are on immunosuppressive drugs or on chemotherapy or on radiation, unfortunately, their neutrophil count actually tends to go down, and a condition actually happens which we call as a neutropenia. And in that condition, we actually do see that excess amount of bacterial infection can actually happen. And that's why you will see that whenever there is a TV advertisement about the immunosuppressive drugs, they always say that as a side effect, bacterial infection can increase. Next, we got the monocyte and the macrophages, and these monocytes are also produced in the bone marrow and enter the blood. After circulating about 8 hours, as we say, they migrate into the tissue and mature into the macrophages. Macrophages are the larger monocytes that reside in specific tissues and also wander around. They are motile, highly phagocytic, and serves as a sentinel cells sounding the chemical alarm when the microbe invades. We also use a particular word for the macrophages. We call them as the professional antigen presenting cell. And that will make much more sense when we talked about the adaptive immunity, where we will see the macrophages will catch essentially the different microbes and then digest them and display the part of their peptides or signature peptides on the surface of them, and they will go to the particular lymphoid tissue and present these foreign antigens to the T cells to activate the adaptive immune response. So we are going to learn about that as well. This is basically what the sounding of chemical alarm means, presentation of antigen. Dendritic cell actually do the similar kind of function, distinguished by long cellular projection, and they are present in the skin, mucous membrane, lungs, intestine, and depending upon where they are actually named in different ways. So say for an example, if they are in the skin, we call them as the Langerhans cells. If they are in the lung, we call this a Kupffer, Kupffer, Kupffer cells. So again, that details is not necessary, but named differently, but these are all dendritic cells. And they are programmed to detect the phagocytose pathogen. So essentially what happens in their long arm, they have receptors. Particularly, we're going to learn about that. These are the toll-like receptors and other type of receptors that can bind to the pathogen and they phagocytose the pathogen. And after processing the pathogen, they display the foreign antigens on their surface to share vital information with the lymphocyte to stimulate an adaptive immune response. This is the same thing that I talked about. The macrophages also do the same thing. So the dendritic cells, as well as the macrophages, they serve as what I said, as the professional antigen presenting cells to the lymphocytes to trigger the adaptive immune response. Other than that, there are also several cells which are the innate lymphoid cells. And most of the cells that we so far talked about, that these are all from the myeloid progenitors. So the hematopoietic stem cells divide into the myeloid progenitor and the lymphoid progenitor. Most of them are the myeloid progenitors so far we talked about. Then we are talking about the ones which are the of the lymphoid progenitor. And these innate lymphoid cells, or ILCs, do not express any antigen-specific receptors. And they lack receptor that responds or remembers specific pathogens. This is unlike the T cells and the B cells, which are the main players of the lymphoid cells, essentially. So there are several types of these. One is what we call as a lymphoid tissue inducer. This facilitates the development of the tissue found in the lymph nodes, Peyer's patches, which are the lymphoid tissue diffused through the intestine. And the others are divided into this innate lymphoid cells 1, 2, and 3. And let's take a look at what essentially they do. See that the ILC1 essentially helps in the macrophage activation. ILC2 basically use the mucus production, vasodilation, macrophage activation, also thermoregulation. That means the fever control and development. And the ILC3s promote the phagocytosis release of antimicrobial peptide and also epithelial cell survival as well. So intracellular pathogen is also detected by the NK cells, and we are going to talk about that. This NK cells is also from are the innate from the lymphocytic progenitor or the lymphoid progenitor and kill the infected cells. We are going to talk about that. So this is essentially all the functions that are mentioned in here as well. So the natural killer cells are really large granular cells destroy the stress of the malignant and viral infected cells. This is an integral part of the innate immune system, but their origin is basically the lymphoid, which is mainly the T cells and the B cells. So basically what they do, they survey the cells all throughout the body by several surface receptors. And then when activated, they kill them by certain mechanism. And we call this an antibody-dependent cell-mediated cytotoxicity. And we're going to take a look at this. So essentially what happens that particularly for the NK cells to function, there are some specific receptor interaction that can happen. So all the cells essentially have what is called a survival receptor and an activation receptor. Now, both of these receptors, you have a positive receptor and a negative receptor. Both of these receptors must be active to make the NK cell function. So what we mean by that if we have the survival receptor on, at that time, the NK cell will actually not kill them. But if the survival receptor is taken off, and along with survival receptor on and as well as the activation receptor on, the cell will actually think that there is something not correct. So it will basically keep the cell under surveillance, but immediately will not kill it. But if the survival receptor is off and the activation receptor is essentially on at that time, it will understand that there is something wrong going on with the cell. The survival receptor is absent and the activation of the NK cell receptor is active. That means that cell is under distress and telling someone to kill it, and it will essentially kill it. And this interaction happens with the MHC, and we're going to talk about this in much later part, particularly in the chapter 32. These are called the major histocompatibility complexes. These are particular long proteins which are present on the surface, and these MHCs essentially can tell, depending upon whether it's an MHC1, MHC2, these essentially are recognized by the NK cells. And the amount of MHC a normal cell actually distributes as a survival receptor will vary. And when they are absent as a survival receptor, those are the cells which the NK cell will think that it has to kill. So essentially, at that time, it also has to get the activation signal as well along with that. So how essentially basically what happens that when the cell the NK cell gets all the correct information that the target cell is infected by a virus, or it is a cancer cell which doesn't have the survival receptor anymore and telling the cell to kill it, it will actually extend some of these nanotubes and there will be an extensive rearrangement of the cytoskeleton, particularly the actin, and it produces a cleft, which we called as an NK cell cleft. And now what will happen that in the NK cell cleft, the cell actually release the lytic granules, which are called the perforins and the granzyme. And these perforin and the granzymes essentially will trigger the apoptotic pathway, which is the program cell death. And the cell will die and the NK cell will separate out. So this is basically the killing of the either an infected cell, mainly virus-infected cells, or cancerous cells, which are not normal or dying cells or infected cells by this process. On the other hand, there is also another method, which we called just right here, antibody-dependent cell-mediated cytotoxicity, or ADCC. Here, what happens, sometimes if a cell is infected by a virus, the cell actually displays these viral antigen as they are as they are dying or in the process of infection, the cell is producing all the viral components. But sometimes there is a system that actually steals a couple of these viral antigens or the part of the virus and displays it on the surface of the cell. This is basically a SOS response for the cell. So these virus antigens are the foreign proteins that are displayed on this, and this can be bound to the antibodies. And when this cell is bound by the antibodies, the NK cells does have the antibody receptor, and depending upon this antibody binding, this can also lead to the same way that it can release the granzyme and the perforin and kill the cell. So this is basically the antibody mediated killing. This one is MHC and receptor mediated killing, essentially, understanding that it is infected. This is MHC mediated, whereas this one is antibody mediated killing of the infection, of the infectious cell. So that is all about the different types of cells, which are the part of the innate immune response. And that would be our part 1 of our chapter 31.